Thank you very much for having me here. This is my first time in Marshall. So I wasn't sure what to talk about. I could say I kind of specialize on queen biology. I did my PhD with Tom Seeley on swarming behavior, then with David Tarpey, the postdoc, on queen supersedure, trying to understand how queens get superseded in the colony. And Andrew asked me to give a talk about queens because it's the right time of year. So I thought about giving this talk on queen management essentials, hoping that at least one of the topics that I will cover are new to you and you can actually apply in your hive. So there are so many different factors that affect queen quality: nutrition, parasites, in particular the varroamide, pathogens, pesticides, genetics, the queen's mating condition, her age, climate change, habitat fragmentation, and last but not least, our own beekeeping practices that are affecting the quality and longevity of our queens. And so I'm going to try to go one at a time and show you some combinations of basic biology, but also some of the research that has accompanied what we now know about those topics. So in terms of genetics, the genetic strain of your colony, of your queen, is a huge factor influencing the growth, the speed of growth, the turnover of that queen, etc. So the race and the genetic strain are very important. She also brings the genetic diversity of her parents and of her own drone partners. So all of those genetics that come from the strain she comes from, so it's Apis mellifera ligustica, or Apis mellifera mellifera, or Apis mellifera scutellata, which would be the African strained. The degree of inbreeding is really important, and so in beekeeping we're trying to avoid queen inbreeding issues because it causes a lot of issues. So increasing genetic diversity by having queens that are well mated is really important because it decreases inbreeding problems. And in particular, inbreeding causes something that you may have heard in beekeeping school, the production of diploid drones, but you may not actually know how that happens. So how many of you have heard of diploid drones? Actually, just a handful, so that's great. In Beekeeping 101, you've probably learned that honeybees are haplodiploid, right? So males come from unfertilized eggs, and that means that they are haploid. They only have one set of chromosomes. The queen lays an unfertilized egg. So drones, the saying is drones don't have a father, but they have a grandfather. They come from unfertilized eggs, and in genetic jargon, that's called haploid. One set of chromosomes that comes from the mom. And then queens and workers, the female caste, comes from diploid eggs. So they are fertilized with a couple of sperm cells, and they finally produce a fertilized egg that can turn into a queen or a worker based on nutrition of the larvae. So those are diploid females. So haploid. So two sets of chromosomes for diploid females, one from the dad, one from the mom. That's kind of the basic knowledge of sex determination in honeybees. But there's a little more nuisance to this. One thing is that honeybee sex is also determined by a gene that's called the sex-determining gene that has 15 different alleles. So this will bring you back to genetics 101 or biology 101. How many of you ever saw Mendelian genetics, little a, big A, in high school, right? And so the big A, little a, or big B, little b, those are what we would call alleles, or the different flavors that the gene can have. So if an egg is unfertilized, that individual only has one allele at that sex-determining locus, because there's only one gene, right? One set, one gene, and so that's fine. That individual is going to develop into what I mentioned earlier, haploid drone. The situation, what's very interesting in sex determination in honeybees is when the egg is fertilized. So that egg, if that egg is fertilized, then that individual is going to have two flavors of that allele, one that comes from the mom, one comes from the dad. If they happen to be different, if the alleles are different, that bee develops into a diploid female, which is the vast majority of individuals in the colony. All of the 99% of the workers in the colony, diploid females and one diploid queen. What's at stake here is if for any reason the alleles are the same, that individual will develop into a diploid drone. So let me give you a better example here. You have a queen, whatever you want to call her, but that queen, because she's diploid, she has 2 alleles at that sex-determining locus. And her flavors are big A and big B, AB. So she is what we call in biology heterozygous, different, right? Homozygous would be big A, big A. Okay. This queen in this particular example mated with 5 drones. And as I said, they only have 1 set of chromosomes, so they only have 1 allele at the sex-determining locus. And in this case, they go from A to B to C, but then she happens to mate with another B here and drone D. So if you do the Mendelian genetics, right, half of our offspring are going to be, well, I guess in this case, a fifth of our offspring are going to be sired by this drone, a fifth one by this one, et cetera, et cetera. And so half of our offspring are going to have this allele, the other half are going to have this allele. And so when you do the Punnett square kind of combination, this one is going to be big A, this one's going to be big B, BA, and so on and so forth. Do you see the combinations here? In the particular event that she mates with a drone that shares any of the 2 alleles she has at the sex-determining locus, that individual, half of them at least, are going to be homozygous at the sex-determining locus. In this particular example, 3 individuals. And so that means that they are going to be turning into diploid, not females, but drones. So the queen is trying to outbreed, going far away so that she doesn't interact with any drones that are relative or related to her, to avoid that by random chance she shares any of these 2 alleles with the drones that she's mating with. So she's trying to increase the odds by outcrossing, leaving far away from her hive, whereas drones nearby, they will stay. And DCAs are closer to their home colony in many ways to avoid spending so much energy going out and back trying to mate. But in these particular scenarios, if you have queens that are inbred, that have higher chance of breeding with related males, then you're going to have too many diploid drones, which causes what beekeepers know as shot brood pattern. So how many of you have ever heard of shot brood pattern? A few of you. So shot brood pattern is if you're going through your inspection and you don't see any sign of disease, and you see that the frame is otherwise looks like it has a steady, clean pattern of worker production, but a bunch of open cells in the middle, like shot brood pattern, that's probably because diploid drones don't ever make it to adulthood. They get cannibalized by workers. They smell differently to the bees, and they get removed by the colony. They know they will not be viable, and so they get removed before they emerge as adults. And so that's why you will see a pattern like this, which is indicative in many ways of queen inbreeding problems, which you should resolve by potentially requeening the colony. So amongst other things, not a whole lot of people have done work on diploid drones, but they've shown that diploid drones have a smaller number or a lower number of wing hooks. The wing hooks are the ones that allow them to connect the fore and hind wings into one mega wing that help them fly faster. And so diploid drones are kind of deformed, and they don't have as much good performance as normal drones. This, by the way, this was done artificially by the researcher allowing those drones to emerge, because if they had not caged them so that the workers had access to them, they would have removed them before they emerged as adults. So we need to avoid inbreeding, and so that means that you need to have sufficient numbers of drones in the mating area, and the mating conditions need to be looked at. So pay attention to the queen's natural mating conditions, especially during this time of year where it's kind of the beginning of our mating season, and you may or may not have drones in your colonies. And of course, we know that the queen doesn't mate with her own drones, but if you're in the vicinity, then your neighbors are probably experiencing, their colonies are experiencing very similar environmental factors, and so their colonies should hopefully also have the same kind of developmental time for drones to be ready. So do you have enough drones during the split time in the spring and during fall requeening replacement? Because if you don't, then the queen doesn't mate with enough numbers of drones, which will kind of help the supersedure or queen replacement process. And what is the quality and or the breed of those drone sources nearby? There's some work that has shown that the further away that the drone congregation areas are from managed apiaries, the higher the likelihood that those drone congregation areas are going to be full of Africanized drones. And so you might want to know what managed apiaries are near your colonies to kind of get a good, or at least a guesstimate of where your queens are going, because that's one of the beauties and difficulties of honeybee breeding, is that they mate naturally out in the open, and we can't really control what the queen does in terms of mating unless we bypass the mating through artificial insemination, which also brings about its own complications. So in the study that we did a few years ago with my student Adrian Fisher, we were looking at the effects of rearing drones in pesticide-laden wax. And so wax that was contaminated with field-relevant concentrations, which is a key word, field-relevant concentrations, so concentrations that actually exist out mostly in large commercial beekeeping operations. And so if we rear drones in wax that's old, that it's lipophilic, keeps sucking up all the contaminants from what we add as beekeepers to combat the varroa mite, miticides, and other agrochemicals, the more we leave these frames in our operations with all of these chemicals on them, the more likely that drones are going to suffer during development. So spermatogenesis or spermiogenesis in drones occurs during development, not when they are born. So they emerge with all the sperm they'll ever need if they're lucky enough to mate. And so if they get reared in wax that's contaminated with agrochemicals, there's going to be a compromise in the viability of the sperm, which means the percent of sperm that would be viable and could fertilize eggs. And so when we rear drones in wax that was contaminated with field-relevant concentrations of amitraz, compared to those that were reared in clean wax, we noticed that the viability of the sperm, again, the percentage of sperm that could fertilize an egg and wasn't dead, was significantly lower than control wax. So that's one of the things that is going on out there. I think one of my take-home messages for this lecture is that queen quality is in part caused by all of these biological and environmental effects on the queen, but also on her drone partners, the one that she's mating with. So that paper came out a few years ago, and it's freely available if you want to read it, because we did it with other pesticides as well, not just amitraz. But so exposure to pesticides during development negatively affects drone sperm viability. The age and the vigor of the queen is also very important when thinking about queen management. You have to keep track of your queen's age. Nowadays, it's not such a big deal to use the international color nomenclature of 5 different colors for 5 different years, because if we're lucky, we can get maybe a 3-year-old queen. But we no longer have the case of a 5-year-old queen. Or how many of you have 5-year-old queens right now? How about about 4-year-old queens? How about 3-year-old queens? Then you start raising your hand. So at most, you might need 3 different colors, but 3 is probably too many anymore. There used to be 5 colors for your queen, so you knew what age she is. But it doesn't matter, as long as you know how old she is. That will give you a good estimate of her sperm viability, or the sperm number in her spermatheca, and I'll show you why. So we know that that queens that are virgin have the sperm storing organ just filled with some kind of like saline solution, some bodily fluid that keeps it basically plump and open. It's covered by a net of tracheae that help keep the spermatheca oxygenated. And when the queen mates, the sperm that she receives from her drone partners goes up some little duct, a little tube up to the spermatheca. It takes about 48 hours for the sperm to migrate up to the spermatheca, and then it looks like this. It's a marbled color. It's not one solid color. It's a combination of like a marbled white and yellow and brownish color. The more brownish and kind of thick the color is, the better mated the queen is. You can do testing of queens by removing the spermatheca. Of course, this is destructive, but if you have many of your queens and you want to test, sometimes you can have something here in between where the spermatheca is filled in a color that looks almost like fat-free milk. And so that will tell you that, yeah, she probably mated with only a handful of drones, and so it wasn't enough to fill the spermatheca with enough sperm. So a lot of people have looked at what is properly mated number look like. How many sperm cells should a properly mated queen have for us to call her properly mated? And that number varies, but it goes between about 3 million and 5 million sperm cells in a recently mated queen. And so for this particular study, they reared a bunch of queens, and I don't remember how many, but let's say 60 queens in year 1, and they allowed all of these queens to mate naturally. They took 1/3 of the queens, they sacrificed them, and they did sperm count of the spermatheca, and they calculated they had about 4 million sperm cells each. They let the rest of the queens go, in little nukes, and then the next year, whoever survived, they took half of those, and so let's say roughly 20 or less. They sacrificed those queens, they looked at the sperm count in the spermatheca, and then they let the rest of the queens go until whoever made it to the next year, then they took everyone out and dissected the spermatheca and did the sperm count. And what they found was a dramatic decrease in the number of sperm just within 1 year. So more than half of the sperm had been used up in a 12-month period by the queens. Naturally mated queens, nothing really difficult or wrong happened to these queens. It's just the natural use of sperm dropped to less than 50%, and then to another 50% of what was remaining the following year. So you can see now why so quickly queens they run out of sperm. By 2 years, they only had about 1 million or less of sperm cells for fertilization of eggs. And it's not a one-to-one thing. It's not a 1 sperm per 1 egg kind of situation. It takes about 3 to 5 sperm cells per egg. And then one of those is the one that fertilizes the egg. So you can see how the queen will run out of sperm rather quickly. Not only does the queen run out of sperm quickly, but she also undergoes, just like we are all undergoing right now, aging, oxidative stress. And so the sperm in the spermatheca is also aging, even as the tracheal net can help manage that oxidative stress with that tracheal net and all of the proteins that are added so that they keep invigorated. Aging still happens. And so a 2- and 3-year-old queen is actually old for a queen. In that same study that I showed you about the queen sperm number, they also looked at the motility of the sperm. And this was really cool. How many of you have ever seen bee sperm under a microscope? Probably nobody, right? I have, because I happen to study bee sperm. But it's incredibly odd looking, because it's almost like a long piece of hair with almost no head. It's just like one long piece of hair. You have to dye it with special dyes to actually see where the head capsule is. It's not like the one that you see in biology class that has the big plump head and a really short wiggly tail. It's actually just one long piece of noodle. And so because the tail is so long, and we don't really know why these tails are so long, they are coiled. And so I don't know if you can see these clusters, but you can see clusters of sperm that are basically tangled together, just like when you have noodles in a pot. They just tangle together, and they move fast in circles. You see them under the scope, and they just keep going round and round and round and round. About 2 years later, in the older queens, the speed of that sperm drops significantly. They become slower, more sluggish. And because she's used up so much of that sperm, the sperm uncoils, and now it's flat, and instead of moving in circles, it moves like this, longitudinally, much slower. And that's what these little noodles, hair-looking things are. Can you believe that? That's a bee sperm. You can't even see the head unless you dye it. And so oxidative stress is happening inside the spermatheca of the queens. That's yet another reason why ultimately we or the bees have to replace the queen, because she is running out of steam, and she's becoming more sluggish, and the sperm in her spermatheca is also more sluggish. So we need to keep records of our queen. Do you have her labeled? Do you know if the queen you have is actually the queen you thought you had, right? Because if you don't keep her tracked, and because supersedure is happening at a much faster rate than before, you may have had a supersedure event in your colony and you didn't even notice. But if you paid close attention, you would notice that the queen that you labeled yellow a few months ago and had this long brownish abdomen is actually not yellow, and it's a short dark brown color. And so now with our smartphones, I would suggest that you take a good picture of your queen as part of your inspection so you know what she looks like. Nowadays it's much easier. So you have to keep track of your queen at all times. And especially, as I said, to keep a vigorous one to avoid this issue of supersedure. As I said, you may have missed the supersedure problem, but if you do inspections every 3 weeks or so, you probably can catch the supersedure act as it's happening. And so as we're playing detective in trying to help ameliorate the queen problem, you want to check out your brood nest area. You're always checking for eggs. But if you notice any cells in the middle of the brood nest area, like these right here, that's a good indication that your colony either already superseded the queen, which means replace the queen, because if you're noticing these cells, then there might be other cells that already have exit holes, which means that the new queen already exited. Or if you're lucky enough, you will catch it before it happens, and you can see, yes, indeed, in the middle of the brood nest area, what the workers are doing is, it's time to say bye-bye to mom, because she's not doing a good job, so we have to replace her. And they actually rear one of these very young larvae from the middle of the brood nest area. They start feeding it royal jelly. They change the orientation of the cell. Instead of horizontal, vertical, and they turn it into a queen cell. So if you see this in your hive, the queen may or may not be there, and that in the case of being there, if you catch it at the right time, if she's there, that's called supersedure. The other event might be if you lose the queen, right? Then they would do something very similar, but the queen is already gone for other reasons. The workers weren't thinking of getting rid of her. She just died of natural causes. Then that would be emergency queen rearing, which is slightly different than supersedure. So In terms of the queen's age and vigor, she is constantly producing pheromones from several glands that together create this very unique bouquet or perfume that makes a particular queen a queen. So in beekeeping, we have the jargon, queen-right colony is the colony that properly functions, has a properly laying queen, and she's releasing amongst dozens of chemicals from the mandibular gland and other glands, this blend of 5 chemicals with these acronyms HVA, HOB, 9-HDA, and non-ODA. This 9-HDA has a mirror image of itself, so that counts as 2, and so there's a total of 5 chemicals that they, in the past decades ago, discovered were the main chemicals that comprised QMP, or queen mandibular pheromone, which, as I said, is produced by the queen's mandibular gland, and it's spread all over her body. But that's not the whole story, and I think I'm going to mention something after this regarding QMP. But imagine these are the little molecules that she's released from the mandibular glands, and she does some self-grooming, so she has all of these molecules on her body. The adult queens that take care of the queen, they're known as the retinue workers. They take care of the queen, and then they do the self-grooming. They move some of those molecules toward the front legs and the antennae and the mouth parts. And then, so some of those molecules are going to get translocated internally in the worker, but then they're going to turn around and do trophallaxis, or food sharing, and they're also going to be sharing the molecules of QMP plus the other pheromones that the queen releases. So if I'm a retinue worker and the queen is right here, I do my self-grooming, and then I turn around and I do trophallaxis and allo-grooming or social grooming with the next layer of workers, and then this worker receives some of those molecules of QMP, she turns around, and so on and so forth. It's kind of like a chain event, such that at any given time, the queen doesn't have to come in direct contact with the worker, or a worker doesn't have to touch the queen directly to know that she's there. She just needs to get enough titers of QMP to know that there is a functioning queen, that the colony is queen-right. So the way that we study mandibular glands is we take mated or virgin queens, we decapitate the queen, and then we take the head capsule and put it on a dissecting plate, and then we open up the head capsule with tweezers and a scalpel, and then take out this tiny droplet-looking organ, which is the mandibular gland, full of, it looks like olive oil inside. It's yellowish and oily in nature, and then you take the glands and do chemical analysis through GCMS, or gas chromatography mass spectrometry, and it tells you that all the chemicals that are found in that gland, and also at what relative proportion compared to the rest. So what we and other people have studied is what effects queen mandibular pheromone has on workers. So we've known for a long time that it has primer and releaser effects. A primer effect is something that happens on and off right away. As soon as she releases QMP, she's calling on her retinue, and the workers come directly and start feeding her, antenating, picking up her pheromone. If you take out the glands and put its contents on filter papers and put the right age of workers, they have to be nurse age, the ones that are really keen on this QMP, immediately they get attracted to the filter paper. It's a really, really potent chemical. It also inhibits worker ovary activation. So remember, how many of you have ever heard that queenlessness leads to hopelessly queenless colonies of laying workers, right? Because in general, the workers are inhibited in their reproduction. So you must be thinking, wait, I was told workers don't reproduce, they can't mate. And that's true, they can't mate, but a certain genetic lines of workers, in the event that the queen is not there, can actually activate a few ovarioles. The ovarioles are those sausage strings that contain the eggs, and can lay unfertilized eggs that turn into drones. So when you see multiple eggs in cells and you keep investigating and you actually don't have a queen, this is the telling sign that you have laying workers in that colony. That's a picture of a laying worker compared to a virgin queen, to a mated queen, actual ovary photo. So QMP elicits the retinue response, and it also inhibits worker ovary activation. Something really cool that happened a couple of years ago was this paper that came out of a lab in Belgium that looked at queen mandibular pheromone and queen-produced chemicals and how it acts to inhibit worker ovary activation, because we had known about the worker ovary activation stuff for a long time in terms of QMP. But as the title says, honeybees possess a structurally diverse and functionally redundant set of queen pheromones. This paper is actually free. And what they did is they found a novel type of queen pheromone that acts in tandem with QMP to inhibit worker ovary activation. So because we have known the chemicals that comprise QMP for decades, people can actually synthesize those chemicals and make them synthetic versions of them and do them, the blend of all five as synthetic QMP. And that's part of, like, when you buy a queen lure, that's basically what you're buying, is a synthetic version of these chemicals of QMP. Or you can do them individually. So the ones that I told you, 9-ODA, non-HDA, HOB, and HVA. So what they did is they took microcolonies or smaller colonies like nukes, and they dequeened all of them. And they waited some time such that the workers realized that they were queenless. And immediately some are gonna say, oh, well, if there's no queen, now I can lay my own eggs. And they start activating their ovaries and laying eggs in these dequeened, queenless colonies. And so those were the controls. They allowed a lot of those, and these numbers are huge. 216, 30% or almost 40% of the workers had developed ovaries. So they're like, well, no queen, then it's my turn. When they gave them, after they had been dequeened, they gave them either the synthetic QMP or individual components of synthetic QMP. It significantly dropped the percent of workers that activated their ovaries, like we had known in the past. What's novel about this experiment is that they also isolated cuticular hydrocarbons from the cuticle of the queen that up to now we didn't know are doing the same thing. They're inhibiting worker ovary activation, cuticular esters and cuticular alkenes. And not only that, they also found that the turgor glands, so the turgor, you know, the last part of the claw, the turgor glands in the queen produce a pheromone that also inhibits worker ovary activation. And that's why they say that it's a functionally redundant set of queen pheromones. They're augmenting the signal to the workers that I'm here and you are not allowed to do any reproduction because I'm the one that lays the eggs here. But if in her absence, then all things go array, and that's what you get, hopelessly queenless colonies. Okay, so moving on to pesticides and how they affect reproductive quality of queens. Just like I showed you the paper on drone reproductive quality affected by being reared in pesticide-laden wax, we did the same with queens. And so we looked at how the exposure to wax, so wax in the queen cups that's contaminated with field-relevant concentrations of miticides and some agrochemicals can actually lower queen reproductive quality and behavior. So what we did is we grafted queens in these cups that were either clean or contaminated with chemicals, and then we put them into cell builders. The nurses reared the queens emerged. They were placed individually in mating nukes We allowed the queens to mate naturally. Once they were mated and we knew they were laying eggs, then we used them for these experiments. We placed them in 3 frame observation colonies like this one here, containing about 5,000 to 6,000 workers. And then we placed the queens caged for a couple of days because you know they need to get accepted by the workers. And a couple of days later, we released them from their cages and we started the experiment. And so we did 2 things. We measured worker retinue size, which is the number of workers around the queen, just like I showed you before. That's a very good indication of how attractive a queen is. The more workers around the queen's retinue, the more attractive her smell is. And so we did that during the day for a minimum of 50 5-minute observations per queen. And of course, we had control queens and experimental queens, meaning the ones that were reared in the pesticide-laden environment. And we also measured queen egg laying rate. So how many eggs was she laying per unit time? Just as an example, this is a very robust retinue of a very good queen that smells like, I don't know, Chanel number 5. This queen right here barely has any attraction, 2 workers around, 3 workers around, no complete retinue. She doesn't smell good at all. She probably smells, I don't know, like Old Spice or something. I don't know. Maybe you use Old Spice. Nothing personal against the Old Spice. They just don't like Old Spice. And so they don't pick up as much pheromones, so therefore it's a kind of a chain of events because then they don't pass around as much pheromone to the next layer of workers. And what we found was quite striking. First of all, in terms of retinue size, here on the y-axis we have the number of attendants per minute, so the number of bees around the queen. And we noticed that for all the chemicals that we looked at, amitraz, fluvalinate and coumaphos, which are the other 2 miticides that we still find in wax, and then the top 2 agrochemicals that we find in wax, which is chlorpyrifos and chlorothalonil combined, the size of the retinue was significantly lower in those queens, for those queens that were reared in pesticide-laden environment compared to the control queens. And very strikingly, what we also found was that the queens that were reared in the miticide groups, fluvalinate and coumaphos and amitraz, had laid significantly fewer eggs per unit time. We didn't see a difference with the agrochemicals in terms of egg laying. So here's the number of eggs laid per 5-minute interval. So what does it mean to lay 1.2 eggs in 5 minutes? That's like not biologically very relevant, right? So I used an extrapolation. Assuming that this queen laid eggs every day, which is not the case during the winter, although in College Station it almost is every day for 365 days. Assuming that the egg laying rate was the same, these control queens would lay about 200,000 eggs a year versus the fluvalinate and coumaphos queens, which would have a decrease of 35% egg laying of only 132,000 eggs a year. And then the amitraz-laden wax queens had a decrease of 43% on egg laying capacity, for only lay about 115,000 eggs. So you can see the repercussions in something as maybe simple, but maybe not as actionable as having chemicals in the wax and how that can affect your queen quality. We also noticed something that I've noticed now in 2 experiments, separate, one in North Carolina, one here in Texas, that rearing queens in pesticide-laden environment increases the mating frequency of queens significantly to a point that it's kind of above and beyond the normal average. And so we still don't understand how that is. So the normal control queens mated with the normal average of 12 drones versus some of these queens that in fluvalinate and coumaphos that mated with almost 20 drones. And so at first I was like, oh, that's weird, but we actually had found the same results in a different study in North Carolina with fluvalinate and coumaphos. Okay, so I talked about pesticides. Now, pathogens and parasites. Our queens, just like the workers, are affected by a lot of honeybee-associated viruses, including those that are vectored by Varroa destructor mites, but they also have black queen cell virus, and they also get STDs. So during copulation, the drone can actually pass along viruses to the queens. So mating is actually a dangerous issue for queens. And they can be so sick from the form wing virus during development that they can emerge with the form wings, and of course these queens would not make it because they can't fly. They also get things like black queen cell virus, in which case they never emerge as adults because they die in the larval state. They also get things like nosema, the microsporidian nosema that causes dysentery, it causes digestive issues caused by two species, Nosema apis, which was the original pathogen of Apis mellifera, and now Nosema apis, which is the original pathogen of Apis cerana, the eastern honeybee, or also known as the Asian honeybee. In areas of the world where they coexist, Apis mellifera and Apis cerana, we have these issues of parasites and pathogens swapping hosts, and that's very similar to what happened with the varroa mite. So in this study, they looked at what happens when queens are affected by Nosema cerana. And so they had control queens, and they had queens that were infected with Nosema cerana. And in what's important about these three graphs here is that those that were infected with Nosema cerana had higher levels of vitellogenin and antioxidative stress responses, meaning that they're working really hard to combat the pathogen internally, which is costly and probably causes a trade-off potentially in egg-laying capacity, etc. And what's also very alerting, to me at least, because we've studied these chemicals in the glands, is that queens that are infected with Nosema cerana have significantly higher levels of 9-ODA, which is one of the QMP chemicals, of 9-HDA, and lower HOB levels. In other words, these queens that are infected with Nosema smell very differently, which means that they are smelling off to the workers, potentially to the drones they mate with if they get infected before, but that's probably unlikely because it would take about 10 days for Nosema levels to peak before they are at its highest, and virgin queens tend to only take about 5 or 7 days in the hive before they roam out to mate. So this is more probably applicable to mated queens. But basically, Nosema infection causes queens to smell very differently, and that probably has a huge effect on how the workers treat the queens. I'm about to finish. I want to talk about the effects of nutrition on queen quality, and we can, there's not a whole lot of work on this area in terms of, there's a lot of work on nutrition, and I could give you a whole 2-hour lecture on what we're doing in our lab when it comes to nutrition of nurses and foragers, but not a whole lot of nutrition studies for queens and drones. There was this one here, quantifying the effect of pollen nutrition of queen egg laying with a new laboratory system that they're actually using quite a lot nowadays. Julia Fine, who runs a bee lab out of the USDA lab at UC Davis, developed this system of their microcolony cages, where she puts 20 to 50 workers, and she has these plates. They almost look like plate readers. They look like comb, but also if you do genetic work, it looks like the 96 well plates that you use for genetics. And then she has feeders and places where she can put the protein feeding, and then she puts a queen excluder material. She has the queen in there, and you can see through the plexiglass the microbe colony of workers tending to the queen and the queen laying her eggs. And then what they can do is take out the plate, and you have the eggs right there, and then they can take them to an incubator and allow other bees to actually take care of those developing bees. And so they tested these micro colony systems. They chose number 3 as the best one for their experiments, and nowadays there's actually a whole lot of science being done with those microcolonies, by the way. And they fed the workers in those cages either bee bread, frozen bee bread, or a pollen patty. And they measured how much of each of the types of foods they ate, and they looked at the size of the hypopharyngeal glands of the nurse bees that were taking care of the queen. But in particular for this talk, they looked at the number of eggs that the queens laid. And what's important, the important take-home message here is that queens laid significantly more eggs when they were fed when the nurse bees were fed the real deal. Of course, the queens are not feeding directly, they're being fed by the nurse bees. The nurse bees are feeding on whatever they were given, which was bee bread or pollen patty, and then they turned that into food for the queen, and the queens that were fed, when the nurses were fed bee bread, were laid significantly more eggs than those that were fed pollen patties. So the real food is always better than the fake food, and it's true for workers and also for queens. I'm not going to go too much into detail about how habitat fragmentation is affecting our colonies, but if we have destruction of natural habitats, especially in colonies that live in rural areas, you were going to see a decrease in the amount of forage available for those bees, which, as you just saw, then has direct implications for even queen egg laying. In terms of climate change, we have a huge issue now with prolonged droughts that don't allow flowers to produce nectar or pollen, and intense rain periods where even though the flowers might have food available, the bees can't go and get them because there's these huge storms, and so they lose these windows of opportunity caused by extreme weather. Nowadays, because this problem with climate change, extreme temperature fluctuations is happening more and more, people are now starting to do a lot of research, bee research, related to climate change. So in the next 10 years or so, you're gonna start seeing a lot more of how bees are affected by dramatic changes in temperature. There's actually a paper that just came out last week out of the USDA lab in Arizona looking at, claiming that warmer temperatures are going to affect overwinter mortality of colonies, and that paper is actually free to download, and I would encourage you to look for it. It was all over the news. Marla Spivak, who runs the bee lab in the University of Minnesota, did a short paper, obscure. She was actually surprised when she saw me quoting this paper because nobody ever paid attention to this. It was back in 1991, I believe. And she incubated queens of 2 different lines at 3 different temperatures once the cells were capped. And so she increased the temperature by 2 degrees Celsius. And she noticed that for both lines, the warmer the temperature during incubation, the faster the developmental time of those queens. And they emerged sooner. So instead of emerging at day 18, they emerged at day 16 for 2 degrees higher, and then at day 15 for 2 degrees higher, which means that these queens are expediting their development and probably not growing as big as they should have had they had an opportunity to develop at a normal time. And so these are very naturally occurring temperatures nowadays. This was in Minnesota, so if we did the experiment again, which I might try in Texas, I wouldn't go for 33 or 35. I would probably go for 37, 39, 41 degrees, which are temperatures that we experience here in Texas during the queen rearing season some years. And finally, our own beekeeping practices. So we need to pay attention to what we do as beekeepers, or I guess most of the stuff that I talked about is relatively actionable. So paying attention to whether you're noticing supersedure cells, or if you see something like this, you see a frame that only has worker comb, and it has sealed worker brood, but then suddenly next to it, in an area there should be worker brood, you see the dome capping of cells. That is indicative of laying workers, or the queen laying eggs that she thinks are fertilized, but she ran out of sperm. She's on her way out, she's shooting blanks now. And so what she thinks are worker destined eggs are actually drone unfertilized eggs, and they are reared in worker brood area. That's a very good indication that you have drone-laying queens. So, and another thing, these telling signs of these exit holes for supersedure or emergency queen rearing is telling you that it might already be too late for you to do anything about your queen except to know that she's probably not there, and you can choose to leave whatever new queen is reared by the colony or take action and change her. Yeah, this is again multiple eggs in a cell. That's a good indication of laying workers. And so you might think, well, I'm going to do an experiment and I'm just going to let these colonies rear their own drones, because at least those drones are going to go out and mate with queens from other colonies. So I am passing along the genes of that queen indirectly through the drone line. But that study was already done, actually. They looked at the traits of drones from laying worker colonies compared to queen-right colonies. And so they dequeened some colonies and they looked at the drones produced by the workers in those colonies compared to the drones that are produced by queens. Every little metric they looked at, body weight, ejaculate volume, number of sperm, concentration of sperm, and the sperm number per milligram of body mass, so how kind of correcting for the size of the drone, all of them were significantly lower by a whole lot in the laying worker colonies. So drones that are produced by workers are much smaller and they have much lower amount of sperm that can fertilize eggs once those drones mate with queens. So it's not a good idea to let those colonies be drone producers. So in short, I hope that you learned something about the biology of queens or drones today that you didn't know before. And to start playing detective in your own colonies might be a good idea to think about requeening every 1 or 2 years, because a young productive queen translates into more populous colonies with higher honey production. In general, there are so many advantages. Colonies headed by first-year queens are less prone to swarming, and if you allow the workers to do the requeening for you and just take your chances, if they fail at trying to requeen in the fall, that can, in the spring, you will prevent a population buildup that can take care of the honey flow. And in the fall, failure of requeening, if the workers decide to do it and not you, can endanger your overwintering chances for that colony. Remember from what I just showed you, the longer you let a colony being queenless, the harder it will be to be requeened. And so that's where you will have laying workers and getting a hopelessly queenless colony. So that's a lot of the things that I just mentioned are just queen management essentials, essential knowledge that will hopefully enable you to become a better beekeeper and a better steward of the queens and drones that you have in your colonies. Incidentally, this is a shout out for our annual, almost annual, we didn't have one last year, but it will be our 7th annual Art of Queen Rearing workshop on Friday, May 24th and Saturday, May 25th at the Bee Lab. It costs $200, and we invite experts in the field, including Your Majesty Sue Kobe, the person that takes all those beautiful pictures of spermathecae, and she does the instrumental inseminations for all of the laboratories. She's actually retired now, but she is a good friend, and she's come to almost all of them. Melanie Kirby is a queen producer from New Mexico. You may recognize Megan Mahoney. She is a queen producer here in Texas. This is Ashley Ralph from Prime Bees. Jennifer Saruda from University of Tennessee, Knoxville came last year, but I don't know if she's coming this year. This is a picture from 2022. And then my students are also instructors in the program. So when registration becomes open, which should happen in a couple of weeks, we will let you know on our Facebook page. So I highly encourage you to follow us on Facebook. We have almost 6,000 followers from all over the world, and that's where we advertise all the new science that's coming out of the lab, all of the workshops and events we're having. If we have any vets, but not veterans, but veterinarians in the audience, we are, or if you know of anybody, we have a free workshop on April 20th at the B Lab for practicing veterinarians or students of veterinary practice that want to learn about honeybee diseases and beekeeping, and that's free on April 20th. But you'll learn about all of these opportunities on our Facebook page, tamuhoneybeelab.edu. So thank you very much, and I don't know if I have time for questions.